be seated in the auditorium so that we are going to begin the session shortly.
आप लगाइए इसको, फिर मैं वहाँ ऑन करता हूँ।
Sorry for bothering you. If you can again speak few words so that we can check that if you're audible offline also.
Hello, Arsa sir.
Yeah.
Yes, sir. Please speak, sir.
Okay, I'm audible.
So you're audible, but not offline, just to me. We are doing it from our end.
Technical problem.
You are audible to all only.
So can you just take your words?
ठीक है sir.
Yes, sir. Sorry for the inconvenience caused, and we have sorted out, and I do hope that sir is quite audible to the audience over here.
तो फिर हम वहाँ जाएंगे ना, अपना वो खोलना पड़ेगा ऑडियो, तो पहले खोल लो उसको, खुला होगा
Sir, now the student may please mute yourself. Thank you, sir. And now we are going to continue with the session, technical session board, day 3, AAE 2023. And we are had the privilege to now introduce the honorable speaker of this session, Dr. Mohammed Azhar. I'm privileged to introduce the honorable speaker of this session, the gathering online and offline. In the world of events, speakers play a crucial role in imparting wisdom and creating a lasting impact. And I'm really honored to now introduce the honorable speaker of the present technical session to the gathering online and offline, Dr. Mohammed Azhar. Sir pursued MSc in Zoology in the year 1996 from AAU, Aligarh, and his PhD in Zoology in 2003 from CSIR KDRI, Lucknow. Presently, he is working as an associate professor, Department of Zoology, ANU, Aligarh, Uttar Pradesh. He pursued his PDF from the Department of Cellular and Molecular Biology, Center of Osteoarticular and Tender Tissue Engineering, University of Nantes, France, during the year 2003-2004. He has keen research interest in molecular endocrinology, bone cell biology, cancer cell biology, and byproducts, predominantly including PUFA and collagen. Dr. Azhar has supervised over 5 PhD scholars and co-supervised over 5 PhD scholars and mentored more than 3 PDFs so far. He has received several research grants from several funding agencies of repute, including the ST, ICMR, UPCST, etc. Further, he has over 74 articles published in several national and international journals of repute having high impact factors. Today, the title of his talk will be Cell Lines as In Vitro Preclinical Test System for Evaluation of Promising Anti-Cancer Agents. With this brief introduction, I request Dr. Azhar to kindly make his presentations. Thank you.
First please. ऊपर आ जाओ। Okay, I'm audible.
Yes, sir. Yes, sir.
Okay, thank you very much for kind introduction. And I am very much thankful to the organizing committee which provided me this opportunity to present my research studies on cancer cell lines. So especially to the Professor Anuradha, and she has been my collaborator in Lucknow, and we have published so many research papers when she was working in Integral University and I was in Lucknow University. So the topic of my talk is cell lines as in vitro preclinical test system for the evaluation of promising anti-cancer agents. Can you move inside?
से हो जाएगा नहीं
So the drug development is a complex and expensive process, as we know that it requires so much complex system like synthesis, isolation, and characterization of the promising compounds, then the preclinical studies, then the toxicity studies, and then the clinical studies. So there is a long way system to perform a study for the drug development. So traditionally, the in vivo animal studies have been used since long for the preclinical testing. And recently, there is the development for the growing interest to use the in vitro model as an alternative of in vivo system or to reduce the number of animals for the drug development. So there are the limitation of the in vivo studies. In vivo studies, we require large number of animals. And as you know that for the estimation of lethal dose or the LD50 dose, we require different groups of the animals for the testing of the different concentration of the compounds, promising compounds. So we make the different groups, and in each groups we require so many animals and for the significance study. So the in vitro study which minimize the use of the animals, and the in vivo study is the costly, time consuming, and clinical trial that may be ultimate fail due to the unforeseen toxicity. So toxicity studies not matching with the clinical trial. So there is the requirement of the substitute or alternate method to the for the preclinical study. So for in vitro studies, there is the cell culture system, primary cell culture system, and cancer cell lines. So in vitro model can be a better replicate of in vivo characteristics while also providing a more controlled and standardized environment of testing. And we can do these in vitro study on specific cell types. If you want to study on the breast cancer cell lines, we can find out the breast. We can find out the cell lines which are related to the particular tissue, like osteosarcoma and epidermal cancer or sarcoma, carcinoma. So we can find out that cell lines of the particular origin which we want to be studied. In vitro models are generally more cost effective, and it requires setup of the cell culture system, some chemicals, and cell lines. Additionally, in vitro models can be performed in more controlled and standardized environment, which can reduce the variability of the results. And in vitro models are also generally considered to be more ethical as compared to the in vivo study, as we require the animal ethics approval from the institutional ethical committee for the study, for the in vivo study. So regulatory bodies are increasingly pressuring researchers to reduce, refine, and replace animal experiments whenever possible, and which may increase the use of in vitro models. So in vitro models can be used to study the mechanism and action of the drugs at the cellular level, at the molecular level, and we can find out the new therapeutic agents at the preclinical level. So the cell lines actually is the culture of primary cells, and when it will be subcultured continuously, it will become immortal. And when it will become immortal, then it is called as the cell line. So there are the different way to develop the cell lines, and cell lines may be finite and indefinite. Finite cell lines, they have the limited number of cell generation, and we can grow in a particular time period. It is a slow growing, 24 to 96 hours time have been taken. So indefinite cell lines, it can be transformed, and it is maybe the cancerous cell lines, and these cells divide rapidly with the generation time of the 12 to 14 hours. And it can be continuously grown generation to generation. So in India, we have the cell repository, that is the National Center for Cell Science, NCCS Pune, and we can find out or we can obtain the cells from NCCS Pune. And there is the list on the website of the NCCS, so we can find out the different types of the cell lines, and we can obtain the cell lines of our interest. And these NCCS Pune providing the cell lines to the researcher in academia and government, as well as the private research institution and industrial establishment in India. So next thing is what kinds of the promising anti-cancer agents. So it may be a different kinds of the promising anti-cancer agent. It may be synthetic compounds. It may be different types of nanomaterials like silver nanomaterials and gold nanomaterials or herbal nanomaterials, nutraceuticals, and different types of the phytochemicals which can obtain from the plant sources. So in our lab, we are focusing on the anti-cancer properties of phytochemicals and Indian medicinal plants, although we have evaluated anti-cancer activity of synthetic compounds and nanomaterials with our collaborators. So why we choose the phytochemicals? The phytochemicals are the natural occurring compounds. They are available in the nutritions, and phytochemicals are largely responsible for the protective health benefits, and they contain the antioxidant components. They are scavengers of the reactive oxygen species and toxic chemicals. Some phytochemicals like flavonols, they mimic with the hormones. They have their affinity to bind with the hormone receptors, and they have least side effects. So we have chosen phytochemicals for the evaluation of the anti-cancer activity. So these are the sources. There are many nutraceuticals, fruits, vegetables. They contain different types of the phytochemicals, and they have the different characteristics like flavonoids, phytic acids, saponin, ellagic acids, phenols, monoterpenes, gingerols, flavonoids, and so many different chemicals nature they have. So when we are targeting about the anti-cancer activity, we concentrate on the two different processes of cancer cells, either inhibiting the proliferation of cancer cell or induction of apoptosis or programmed cell death in the cancer cells. What is the apoptosis? Apoptosis is the programmed cell death, and when normal cells undergo in the process cytoplasmic changes, nuclear changes, DNA fragmentation, then mitochondrial potential changes, and lastly, it forms the apoptotic bodies and cell death occurs. So the purpose, when we are testing an anti-cancer compounds, so we target to inhibit the proliferation or induce the apoptosis in the cancer cell. So this is the mechanism how the apoptosis have been induced by these phytochemicals, either extrinsic pathway, the compounds they binds with the receptors of the cancer cells, they induce the caspases and further induction of the apoptosis. Either the compounds they work with along with the intrinsic pathway by pro-apoptotic markers or anti-apoptotic markers, and they induce the mitochondrial changes, mitochondrial potential changes, and they induce the cytochrome C and further caspase system and apoptosis. So for our study, we have selected 3 different phytochemicals, piperine, naringenin, and eupallatine, and the markers of the cell proliferation and apoptosis. So first marker is cell viability test, MTT assay, then nuclear fragmentation assay, ROS activity. ROS activity means that the reactive oxygen species activity, DNA fragmentation test, mitochondrial membrane potential, cell cycle analysis, and caspase 3 activity. So these potent markers we have selected from the pathway of apoptosis. So in our first study, which we have done on the piperine, this is the chemical structure of piperine, and it is present in the black paper and long paper. And what we have done, the first we have treated the cells with the different dosage of piperine, different 5 doses, 25 micro-m, 50 micromole, 100 micromole, 200 micromole, and 300 micro. And it is visible clearly that how these piperine changes morphologically the cells. And then we perform the MTT assay for the cell viability, and in the increasing concentration, the cell viability of the cell lines is decreasing. KB cell lines is decreasing. So further, we have tested to the ROS activity in the cells, and for the ROS activity, we have done the DCFHDA test. This is the dye which binds with the reactive oxygen species and give the fluorescent sense. So in KB cells, we have selected 3 different doses, 50, 100, and 200 microgram, and it is clearly that this compound that 50 microgram, 100 microgram, and 200 microgram, with the increasing dose, it increase the reactive oxygen species in the cells. So further, we have tested the mitochondrial membrane potential by using the JC1 red dye. And when the potential is increasing, the JC1 gives the green fluorescent. So with the increasing dose, the green fluorescent is increased, and it shows that the mitochondrial membrane potential is altered with the dose-dependent manner. Let me change.
Next.
Okay. Next slide. Then we have performed the cell cycle analysis. And in cell cycle analysis, we find out the different stages in the cell cycle like apoptosis, G0, 1 phase, G2 phase, and S phase. And we found that the cells are arrested in the G2 phase and it is not going to the mitotic phase. So, and apoptosis is increasing with the dose of the piperine.
Next.
Then we have selected a potent marker of apoptosis, that is the caspase, and we have done this activity by the caspase measuring kits available in the market. So the caspase activity is increasing in the dose dependent manner in the cells. So another compounds, phytochemicals we have taken, that is the naringenin, and we have tested this compound in the human epidermoid carcinoma cell by using the Ross generation and cell cycle arrest. So this is the structure of naringenin, and it is found in the different types of the fruits and herbs. So further, we have tested the cell morphology with the different dose of the naringenin, and you can observe that the changes in the morphology of cells. Then we have done the same test, MTT test, for the cell viability, and dose dependently, the MTT cell viability is decreasing with increasing the dose of naringenin. For the nuclear changes or apoptosis changes, we have found that the naringenin increasing the apoptosis in the cells by changes of the nucleus. So further, we performed the Ross generation assay by DCFHDA test and photomicrographs showing that the intracellular Ross generation inducing by the 100 microM, 300 microM, and 500 microM of the naringenin after 12-hour incubations. And these values are expressed in the percentage increase and decrease. Further, we tested the mitochondrial membrane potential in A431 cells, and you can observe that the green color is increasing. So the mitochondrial membrane potential is decreasing with the increase of the dose. And DNA fragmentation test by electrophoresis. So it is observed that the dose-dependent laddering pattern in the DNA fragmentation in this slide. Further, we tested the cell cycle analysis, and we found that the naringenin with the increasing concentration representing or increasing the apoptosis in the different dose. And further, this is the caspase 3 activity. So these are the markers which we have taken from the pathway of apoptosis. And further, another compound, eupalatin, we have taken, and the similar test we have performed for the test. So our study suggests that these phytochemicals significantly induces the apoptosis in cancer cell via both extrinsic and intrinsic pathway of cell apoptosis. Further, the studies are needed to elucidate its potential molecular mechanism as well as clinical significance. And this in vitro test system, which we have performed for the study of these phytochemicals, it can be used to evaluate the anti-cancer activity of different types of the promising anti-cancer agents. And also the in vitro test system reduce the use of animals, which for the preclinical studies of the anti-cancer agents. So the in vitro system, these are the research papers which we have published from our lab. And this is the research teams which perform these work at the Lucknow University, and we acknowledge the grants from the different funding agency. Thank you very much.
I'm extremely thankful, sir. Hope I'm audible. Hope I'm audible to you, sir.
Yeah, you are audible.
Thank you, sir, for enlightening us with your nice and elaborate talk. Now the forum is open for the question and answer session. And if there are any queries from the audience here, we would like to pick up some. Or else we'll move ahead with queries from some of our attendees who have been attending this event online. So with due permission from you, so we pick some questions for you, sir, for you.
Yeah.
There are some basic questions, sir. Well, I'll pick some questions from some anonymous attendees also. What is the basic difference between a primary and a secondary cell line?
Primary cells are the cells which are directly taken from the animals, and then it is cultured in the cells. And secondary cell lines, when we subculture the cells, and then it will become a cell line. So it is called as the secondary cell lines or immortal cell lines. Sometimes it's become the immortal cell lines.
Sir, I have a query also. Like which cell lines or cell cultures have longer lifespan, whether it is primary or secondary cell line, and with whom it is better to work with? So we go for, so is it better to work with primary cell lines or secondary cell lines? Or what
So
criteria?
it depends on the study. It depends on the study. So if we are studying for the problem which are related to a tissue, so we can use the primary cells. And but the primary cells are not, we cannot culture them generation to generation. So mostly we choose the infinite or continuous cell lines or secondary cell lines for the study. So they have the viability and we can culture them one generation to another generation. So we subculture them more and more time.
Yes, sir. Then some more basic questions from my end also. Like while we are working in a cell culture lab, there are many ways or many options to get our cells or cultures getting contaminated. So what are the ways or what are the causes of cell contamination and how can we avoid the same?
So for the
My question, sir?
yeah, you are audible. Yeah. So you are asking about the contamination.
Yes. Source of contamination and then how we can minimize the same. If you can guide us in that direction.
So we have to take the precautions. So those precautions which are already the standard precautions, to wipe up the hands and wipe up the instruments with the alcohol or ethanol, and to use the first, when we enter into the cell culture lab, we have to use the sanitization method, so sanitize them. And previously we have to be sanitize all the instruments and the workplace and the CO2 incubator for the proper functioning and to prevent the contamination.
Thank you, sir. And one last question from my end. That is there any alternative way that without a CO2 incubator, we can also establish a cell culture lab?
So there is no alternative of CO2 incubator. We have to CO2 incubator for the providing the condition of the requirement of the growth of the cells. As in our body, there is the regulation of temperature for the cells, and as well as the requirement of the nutrition, as well as the humidity. So the CO2 incubator is a system which provides the condition like concentration of carbon dioxide in and the humidity. So it is required for the cell culture, as well as the temperature,
Yeah, sure, sir. Thank you, sir, for this nice interactive session. And yeah, we have one query from one of our esteemed speaker, and I'm so glad and proud to announce that Dr. Tabina Hassan from Saudi Arabia has been attending all our sessions throughout, and ma'am, we are extremely thankful for you. And I would like to take a question for you, sir. Do we have any good cell line encyclopedia that you would recommend for cancer cell line? This is our question, sir. I request you to please address the same.
What are the what is the question? Please repeat your question.
Sure, sir. I'll read out the question again for you. Ma'am,
Different
wants
cell
to know,
lines?
do we have any cell line encyclopedias? I mean, she's wondering if we have some sources from where we can easily find out that which type of cell lines are recommended for which type of cancer, if I'm not wrong.
So we can find out the different cell lines available in the list of the NCCS Pune. And there is a list of the different cancer cell lines and the non-cancer cell lines also, and normal cell lines. So the tissue which we of our interest, or the cancer of which of our interest, we can find out the same origin cell lines from the repository NCCS. And there is a process to be registered with the NCCS Pune. We can go at the website of NCCS Pune, we registered, and then we can order and they will supply.
Thank you, sir. I do hope that ma'am might have received her answer. And now with that question, we have came to the end of this technical session 1, AAE 2023. And I have been finding in the chat box appreciations, words of appreciation being flooding over for this particular session. And I have some couple of questions, if you permit, I will take for you. Should I? Some more questions? Am I audible to you, sir?
Yeah, you are audible.
Sir, we have 2 more questions. If you allow me, I will take the questions also.
Okay. So you.
These are the questions from one of our attendees, Priya Chaudhary. She has been attending us online, and she wants to know that what are some of the major organelles in human cells? It's such a basic question, but might be if you love to answer the question.
Repeat the question, please.
What are some of the major organelles in human cells? She wants to know.
What are the major
Organelles.
Organism. So there are the human cell lines we can find out, the different animal cell lines. Even in India, we have the repository in the NBFGR to find out the cell lines of the fish origin also. So different cell lines are available. Actually, there is the echo in your
Oh, why?
voice.
Okay.
So there is a problem to be here.
So now am I audible to you?
Yeah.
Okay, sir, with that questions, we come to the end of this technical session, 1, day 3 AAE 2023. And I thank you, sir, for accepting our invitation and this event with your presence. And I request you to be online for as we are also continuing with our valedictory ceremony. Sir. Thank you.
Okay, thank you. Thank you for.
Thanks for this nice elaborated session.
Mm.
Warm good afternoon of the day to all the dignitaries, delegates online and offline, my dear colleagues, and my dear student friends. With due permission from our Honorable Director AIP, AUUB Lucknow campus, Professor Dr. Anuradha Mishra ma'am, we are now at the verge of starting our valedictory ceremony of the 3-day extended international conference come workshop on alternatives to animal experiments, exploring imminent possibilities in preclinical drug discovery using computational tools, organized by MIT Institute of Pharmacy, MIT University of Uttar Pradesh, Lucknow campus during 1st to 3rd November 2023. The need of alternatives to animal experiments in preclinical drug discovery is driven by ethical concerns, cost, and time savings, improved human relevance, reduced variability, environmental impact reduction, and increased regulatory acceptance. By embracing computational tools, we can create a more ethical, efficient, and sustainable approach to drug development. AAE 2023 has been an impressive and thought-provoking conference and workshop since the last 3 days, based on one of the most up-to-date themes aimed particularly at students, PhD scholars, educators, researchers, and professionals in the fields of pharmacy, life sciences, biomedical research, and related disciplines. This event was predominantly designed to explore, promote ethical, cutting-edge alternatives to traditional animal experiments, fostering a deep understanding of the latest advancements in non-animal research methodologies. It was also being focused in providing hands-on training to the attendees on handling different freely available computational tools that they can finally use in several drug discovery and development processes, including autolog and Swiss alien. To grace this event, we have among us one of the most eminent personalities as the honorable chief guest of this particular ceremony. And may I now have the privilege to introduce the honorable guest of today's ceremony to the gathering online and offline. Dear audience, we have Dr. Sandeep Chaudhary, Associate Professor and HOD Department of Medicinal Chemistry, OMC Lab, Naigar Rai, Bareilly, Lucknow, as the chief guest of today's valedictory ceremony. Dr. Chaudhary is presently working as an Associate Professor and HOD of the Department of Medicinal Chemistry Laboratory at Naigar Rai, Bareilly. He pursued his PhD from the Division of Medicinal and Process Chemistry, CSIR, TDRI, Lucknow, and has got triple PDF during 2010-2012 from MCRF Institute of Microbial Chemistry, Tokyo, Japan, 2009 to 2010, the City University of New York at Hunter College, New York, USA. During 2008 and 2009, the City University of New York at Hunter College, New York, USA. He has been also awarded with several awards from reputed organizations, including BIFA in 2016, Outstanding Faculty Award by BMCL, Certificate of Outstanding Contribution in Reviewing Elsevier, Amsterdam, DST Serve, Young Scientist Award, Department of Science and Technology, are the few to be mentioned over here, and the list is continuing. He has over 10 years of teaching experience at Naigar Rai, Bareilly at UG and PG level, and Dr. Chaudhary has been bestowed with 2 ongoing sponsored projects and 14 completed projects so far. He has total 75 research publications in SCI journals with H index 19 and I-10 index 38, and total citations 930 more. He has presented over 56 invited talks at different national and international conferences and workshops. Moreover, he has supervised 4 postdoc scholars and 10 PhD scholars so far. May I now have the privilege to welcome Dr. Sandeep Chaudhary, sir, on the stage and bestow his words of knowledge to the audience, both online and offline. Sir, please.
Good evening, everyone. Thank you very much for giving me opportunity to visit Amity University. I would like to thank the chief patron of this conference, Dr. Ashok Chauhan, sir, patrons as well as co-patrons, chairpersons, conveners, and all the organizing secretaries. So it's privilege to visit this beautiful campus. Reason being, I was earlier visited this campus in 2017 when Pro VC was Professor Ori, and I joined here as an expert member to accredit this for BTech examinations through IIT JEE at that time. So at that time, I was serving in Malviya National Institute of Technology, Jaipur. So recently, I moved to this place, permanently moved to this place. So now I will like to give you a brief introduction about the things. As organizing secretaries told me that it is basically a drug discovery based conference. So I should plan, and most of the audience are students. So I planned something, a very basic concept. Maybe if I have been invited in future, I will give you brief introduction about the work which I was carrying at which I had carried out at MNIT as well as currently at NIPER Raebareli. So I will be talking about a basic concept of drug discovery, an overview of drug discovery, what a medicinal chemist basically thinks about to make a new molecule, which can be going from bench side to bedside. So excuse me. It's not moving there.
The display you can hold up.
Okay. So, as you know, so what are the various stages of drug discovery? It actually proceeds much through several stages in order to produce a product that should be safe, efficacious, and has passed all the regulatory. So a molecule which is prepared in laboratory in the bench, which could later on goes to the bedside, it requires several stages and it requires several years. So a drug discovery means a hit or getting a lead molecule, and that if underwent some kind of formulations, then goes to after doing PD as well as toxicity studies, it goes to preclinical and clinical studies. Okay. So, drug discovery, actually the synthesis of molecule, preparation of molecule in the laboratory requires a long time, 5 to 7 years, and drug development, which involves the preclinical and clinical phases. So these club together, you can say that a molecule becomes a bedside portion when it passes through around 14 years. So it's a process.
Sorry to interrupt you,
no,
sir. The PPT is not being shared on the online, so
Okay.
you just give it.
No. So the process of drug discovery actually involves the identification of lead and its target, synthesis, characterization through various spectroscopic techniques, screening, and assays for therapeutic efficacy of lead. Once a molecule has been identified, lead molecule, and then it goes to the drug development stage, which is prior to clinical. So average time required to bring a drug to the market is around 12 to 15 years, and average cost is 800 to 1,000 million dollars. So a drug discovery and development process is a progression from target to and leads to drug to products. So altogether, if you see the chart over here, which actually gives you the clear picture of the drug discovery and development process, where targets and lead are being designed or fulfilled by medicinal chemists. And then when a drug has been, or lead has been confirmed, then it goes to the preclinical and clinical studies. So these are the various phases. So target selection, target to lead, lead to candidate, candidate selection to first time in human, then POC, POC to phase 3, phase 1, phase 2, phase 3, and then to the launching. So altogether, it requires a almost 15 years. It's the same thing. So a good laboratory practice and good clinical practice, good manufacturing practice, everything is required to make a molecule which you called a medicine, actually. So any pharmaceutical company or any institute, if identify any molecule that it is a lead molecule or it's a drug-like molecule, so they actually first file a investigational new drug patent, which is application is the first step for any pharmaceutical company or any government or private institution on their journey to get a new drug to market. And after that, when it passes through phase 1, phase 2, and phase 3 clinical trials, it is also again registered as new drug application. And through that process, and all these processes required GLP, GCP, and GMP processes. So filing of patent is very important, and you might be knowing that when you file a patent, it is valid for 20 years from the filing date. And since drug discovery process is 10 to 14 years, it means that whatever molecule you decided to be a lead molecule, that becomes a drug molecule within the time period of patent. So how to find the right molecule? What should be the approach of a medicinal chemist? What should be the approach of an organic chemist to plan a molecule in the laboratory, which can be put in the next to your bedside in future? So it all started through chemistry knowledge, where you prepare a bunch of molecules, I should say thousands and thousands of molecules. From there, screening in vitro, in vivo screening, toxicity studies are being carried out, pharmacological and safety assessments are being carried out. From there, if you imagine 10,000, it reduces to 1,000. And later on, when you talk about all the in vitro, in vivo, it comes out to be only 10 molecules. And then after further formulations and further submissions, and then you find out only one molecule. So after preparing 10,000 molecules, you end up with only one molecule, which shows a drug-like properties. So this is again the same process which I told you now. So how to prepare a potent drug-like lead, or you can say hit molecule? The very important thing is that medicinal chemists should think of at molecular level, or I should say at gene level or protein level. So which gene is actually responsible for curing or for causing the disease? And of course, what is the role played by the particular segment of the protein, which is responsible for causing some error, some disease in the body? So based on that, literature search through SciFinder and various online sources. So we identify, or we means medicinal chemists identify or prepare several molecules, and then screening of the molecules are being done, and then based on the activity profile getting from screening, we identify a lead molecule which shows a drug-like property. It should follow the Lipinski rule of 5. You might have studied in your B.Pharm classes. And from those molecules, potent molecules, we perform several PK, PD, toxicity studies. And then we say that, okay, we have out of several, we have 1 or 2 potent lead type molecules. So what should be target? How target should be selected? Whether it is established, it's a new. So we have to think of, since many of the targets are known, whatever disease you are doing, listening here nowadays, there are several diseases whose targets are still unknown. So chemist or biologist both work together to identify that particular target, and then they plan accordingly to make a particular molecule in the laboratory, which is not an easy task, and it requires several years to establish a particular molecule for a particular disease. So how to find the drug target? So genome sequencing through which a particular selection of protein which is actually causing a disease in the molecule in the human patient. So biologist, chemist, they all investigate these things, and then after that, and what is the pathology of the disease? What is the at the gene level? What is the scenario? And based on that, what is the biological target? So chemists try to find the molecule. Biologists try to give us the target. Based on that, a particular target as well as particular molecule or particular area has been decided. So screening can be your random, non-random, or cross. Random means no intellectualization, and assay are done without structural things, structural regards. Non-random, which you call targeted or focused screening, and it is a more narrow approach, and this is compound having a vague resemblance to weak active compound uncovered in the random screening process. So whenever a hit against a chosen target will interfere with other related target, this is the process of cross screening actually. So what are the type of screens we are actually thinking? If the compound bind to cell surface receptor, we call it, we measure that is through binding assay. And if that binding evoke some cellular response, that is measured through functional assay. So what are the different approaches for drug discovery? So what is the nature of sources? Chemical sources, rational approaches, molecular modeling, combinatorial chemistry, biotechnology, bioinformatics, preclinical studies, and then clinical studies. So these are the various phases through which a molecule is, fate of a molecule is decided. So I am discussing one by one. Nature of source. Plant is a rich source of bioactive molecules, and you know most of the drugs are actually derived from nature. Nature has immense potential. A lot of things are hidden, still hidden. Although a lot of work has been done, you might have studied, you might have reading in your textbooks, in your literatures, and most of the drugs are also plant derived. Microbes are rich source of antimicrobial drugs, like streptomycin species has been the source of antibiotics. Marine environments are potential source of several new bioactive agents. Arabinose nucleoside are discovered from marine invertebrates. So these are the various sources from where you can find a potent molecule. So before considering any molecule for particular type of activity, we do virtual screening. And virtual screening, you all know that we have well-established docking software you might be knowing, you are using. Some are ligand-based screening you are doing, structure-based, and target-based drug design, or these are all company. And In vitro, in vivo studies are being carried out through these assays, for which bioavailability is one of the important factor which governs that your molecule. If a molecule doesn't reaches to a particular target, it means that even though it has the potential to become a drug, but if it doesn't reaches to the particular target, it means that it will not show activity. So reaching molecule to the target is very important. So for this, several formulations, pharmaceutical scientists are doing formulations, and they make a formulation of that particular drug molecule so that it reaches to the particular target site. So everything started through organic chemistry. Since, as you know, molecules are being synthesized by organic chemist. So organic chemistry, the knowledge of basic organic chemistry is very important to become a medicinal chemist, I should say, to all the future generations. So if you have a clear concept of organic chemistry, you have a clear concept of medicinal chemistry, which you are getting right now in MIT Institute of Pharmacy. So once you have acquired both knowledge, you can become a good medicinal chemist in future. So I mean to say, basic utilization of basic organic chemistry, its synthesis, how a molecule is being synthesized, what is the route of synthesis of particular molecule, how you can introduce chirality into a molecule, how enantiomers, diastereomers are being synthesized. So knowledge of basic organic chemistry is very important. So I'm just skipping because there is a shortage of time. So when a molecule reaches to phase 1, phase 2, phase 3, so we will discuss some aspects, what is actually being studied during these phases. So, and what are the risk and benefit assessments are there associated with that. We will discuss this also. What are the different pharmacodynamic, pharmacokinetic, as well as toxicity studies associated with the particular molecule, considered to be a hit or lead molecule. What are the regulatory guidelines, quality requirements? We all discuss in upcoming slides. So suppose you want to do some carcinogenicity test studies. So as you know, it is expensive studies. It requires 2 years of dosing, requires rat and mouse. So all these phases, so it requires an extensive infrastructure. As I told you in the very beginning, that a drug discovery after 15 years of time, investment and money, it altogether requires $800 to $1,000 million. So, so overall, it's an iterative process. Medicinal chemistry, biological chemistry, biological phenomena, in vitro, in vivo, pharmacokinetic, pharmacodynamic. Based on all these studies, we select a molecule which you call a drug candidate and which is being ready for testing in man. So what are the chemical sources? As I said, one of the best way is laboratory, synthetic laboratory, and then some inorganic sources, some mineral sources, and also combinatorial chemistry. It was started early 90s, but later on it was dropped down because there was issue with the purity of the compound. So combinatorial chemistry was actually giving us a bunch of molecule in less number of time, but later on it was dropped. Nowadays it is rarely being used. Maybe industries are using it. So different types of combinatorial synthesis, split synthesis, peptide libraries. As you know, there are several peptides which can also be acting as drug. So peptides, as you know, these are nothing but the amino acids. So they are also playing a very important role. Nowadays, synthesis, peptide synthesis through peptide synthesizer in laboratories are being carried out for particular disease also. So I think I am skipping. what are the technological approaches? So identification of target is being carried out through the deep study of genetics, molecular biology, bioinformatics, X-ray determination and NMR spectroscopy are the tools for the structure elucidation of the molecules. Molecular aided drug design, as you know, softwares are being used. Different docking softwares are being used to assess whether that particular molecule is suitable for docking into the receptor site of that particular active site of that particular receptor or not. And then computer aided screening is also being started, synthetic chemistry. And once you identify a molecule, then you go for preclinical. So let me skip this part. Yes, so preclinical stages is up to here. And then once you test the molecule in phase 1, phase 2, phase 3. So what are the actually what are the targets in each phases? I will discuss in upcoming slide later on. I am skipping because of paucity of time. So in phase 1, there is actually no blind screening. The number of subjects are actually 20 to 40 to the maximum 50. And it is being targeted ones are healthy volunteers. And sometimes patients are exposed to drug one by one also. And then what are the associated members carried out by qualified clinical pharmacologists and trained? So phase 1 clinical trials are carried out especially by clinical pharmacologists and trained physicians. Physician means doctors. And dose is given in cumulative manner to achieve the effective dose. So purpose of the study is the measurement of PK, PD, and emphasis is on safety and tolerability. In phase 2, the next target are 100 to 400 patients or volunteers according to specific inclusion and exclusion criteria. More or less physicians, trained investigators, and purpose is to establish therapeutic efficacy of drug dosage regimen and ceiling effects in controlled setting. And tolerability and pharmacokinetics are studied as phase 1 extension. In phase 3, the target is now increased. It is the number of subjects now goes to up to 3,000. And all physicians are basically doing this study. And purpose of the study is to establish value of drug in relating to existing one. So a molecule to medicine is a big journey. It requires collective efforts of synthetic chemists, analytical scientists, large-scale pilot plant people, all physicians, all pharmacologists, pharmaceutics, everything is involved. Toxicologists, everyone is involved in studying this. So in lab scale, when we find a lead or hit molecule, we do synthesis on 10 gram to 100 gram. But later on, when the molecule becomes a drug-like molecule, doing the same reaction on a large scale is difficult. Many of the reactions failed, and it is essential for the company to establish particular method or particular procedure to do the same reaction which you do in laboratory. So same reaction performed in laboratory is totally different, which is performed on industrial scale. So once a molecule is considered as drug, which molecule, there are some regulatory authorities. Some of the key regulatory agencies are FDA, EMEA, and MHLW. They seek regulatory inputs during this phase of development to ensure on track with phase 3 plans. The role of regulatory affairs department in every or in any pharmaceutical company is mainly the licensing for communication between the clinical matrix team and the approving agency. So not all regulatory authorities give advice. Actually, in general, interaction with FDA are more acceptable, both informal dialogues as well as more structured regulatory scheduled meeting are done actually. More difficult to obtain scientific advice from the EMEA. And as I shown you, these are the some regulatory authorities. Similarly, there are many different authorities around the world. Traditionally, they have had different requirements regarding the type and amount of studies and other information required to assure the best quality, safety, and efficacy of the drug. Majority of the authorities are listed over here, like Food and Drug Administration of US, Ministry of Health, Labor and Welfare of Japan, European Medicine Agency of Europe, International Therapeutic Goods Administration of Britain, Health Canada of Canada. So, to get a drug molecule, to make a molecule from bench side to bedside, a collective effort of medicinal chemist, or you can say organic chemist, pharmacologist, pharmaceutics, toxicologist, and those involved in metabolism studies are a dedicated study is essential for making a molecule to a drug. So drug discovery is a lengthy process. It is not a one-day job. It requires extensive effort, maybe effort of many researchers involved over the studies. So today I gave you a basic intro about the drug discovery process. Although you might have studied it, I just gave you the glimpses. I didn't talk about my work, which actually I am doing over the last 2 decades. Maybe if MIT University gives me another opportunity in future, I will discuss my work in detail. I have a small team. Earlier, I was in MNIT and MNIT Jaipur. And in 2017, I visited this campus as an expert, as I told you, for accrediting this college for taking BTech students through through IIT JEE and CSAB examination. And these are my teams. I joined in Naipur Library in 2021, and another team has started maybe next. And these are the various funding agencies so far, as Dr. Lucy told you, that I had been fortunate enough to deal with plenty of sponsored research work through national as well as international agencies. Few are South Africa, Indo-Slovenia, Indo-Russian. One of the project, this one was funded by Russian government, finished in 2021-22. Recently, I am handling one ICMR and one CRG is about to finish. So, thank you very much for your kind attention. Thank you very much.
I would request you to get on the stage, sir, because I will now request the honorable dignitaries of state to kindly join us on stage to felicitate our chief guest of this ceremony, Director HR, Adita Ma'am, and our own beloved Director AIP, Professor Dr. Anuradha Ma'am. Please join us on stage for felicitating the chief guest of this ceremony. I would also request the organizing secretary, co-fellows of mine, is to be on stage for the felicitation process. Dr. Rahul, Dr. Yasir, and Dr. Sathapis.
Basic. It was basic. I didn't talk about any life.
I request all our dignitaries to join hands to felicitate our chief guest of today's ceremony, Mr. Santa, and the Honorable Agricultural Mafia, and thanks to the Honorable Chief Guest of today's ceremony, because here at Agricultural we believe that gifting someone a plant is blessing them with a whole new life. So may you long live, sir. And thank you for joining us for this particular session. Thank you, sir, for taking time to speak to our attendees online and offline. And now I will take the privilege to invite on stage our own beloved, respected director, AIP Professor Dr. Anuradha Mishra ma'am, to present the overall report of this 3-day extended international conference and workshop AAE 2023 to the audience, both online and offline.
Good afternoon and warm welcome to this last session. That is, we are going to complete the 3 days workshop conference. That is your international conference. With that, I just want to greetings to all my directors, Director HR and Dr. Sandeep sir for joining here. Dr. Professor Asita ma'am and my other dignitaries and my colleagues and friends. And dear students, those who have joined here offline as well as online, thank you so much for participating in this conference. As we have almost accomplished this 3 days extended international conference cum workshop on alternative to animal experiments, exploring imminent possibilities in preclinical drug discovery using computational tools in 2023. Organized by Amity Institute of Pharmacy, Amity University, Uttar Pradesh, Lucknow campus during 1st November 2023 to 3rd November 2023 on hybrid mode. It is my honor and pleasure to give the closing remarks of this grand event as the convener of this event. This 3 days international conference cum workshop was held on hybrid mode with the major highlights being a workshop on handling free software related to molecular docking such as Autodoc, Swiss, ADME, conducted by 2 faculty experts, which was conducted today morning itself. From our own department, including they are Dr. Rahul Kumar Maurya and Dr. Mohammad Yasir. And the oral presentations competitions was also conducted for abstracts received from both review and research sections, in which prize money for winners will be presented at the end of this valedictory ceremony by our honorable chief guest. Participation was made online for first 2 days and choice for attending the workshop on day 3, online or offline mode was provided to all our participants. That is on the 3rd day. With great pride and joy, I must confess here that AAE 2023 has reached its objective to a larger extent, that is, to prohibit non-productive animal cruelty via experimentations and understand the value of the alternatives for manifesting research and to carry out non-animal testing methods such as computational and to reconnect with 3R principles, that is, refinement, reduction, and replacement among the research communities across the globe. The technical session was highly rich and interactive as we were blessed to have speakers from versatile organizations of repute of both national and international. The honorable speakers of the technical sessions of day one were Dr. S.K. Rath, Chief Scientist, Toxicology and Experimental Medicine, CSIR, CDRI, Lucknow. Good afternoon, sir. I just break here. I welcome our honorable Deputy Pro Vice Chancellor, Sir, Wing Commander Anil Kumar sir joined here. Sir, we are really blessed. Just we have started the sessions. With your permission, I want to continue. Then Dr. S. Anubhumani, Principal Scientist, React Division, Ecotoxicology Laboratory, CSIR, IITR, Lucknow. Dr. Tabinda Hassan, Associate Professor of Human Anatomy, Histology and Embryology, College of Medicine, Princess Noora bint Abdul Rahman University, the Kingdom of Saudi Arabia. As the honorable speakers for day two, we had Dr. N.C. Misra, Health Sciences Manager, Defense Health Agency, University of Utah, Germantown, Maryland, US. Dr. Vinod Tiwari, Associate Professor, Department of Pharmaceutical Engineering and Technology, IIT BHU, Varanasi, Uttar Pradesh. Dr. Mukesh Nandev, Associate Dean, Research and Consultancy, and Head Department of Pharmacology and Pharmaceutical Biotechnology, Division. University, New Delhi. Today's session was graced by Dr. Mohammad Arshad, Associate Professor, Department of Zoology, AMU Aligarh, and HOD, Department of Medicinal Chemistry, Lab, Naib Primary, Dr. Sandeep Chaudhary, sir. Dr. Sandeep Chaudhary, sir, has also joined as chief guest of today's valedictory ceremony, AA 2023. I am honored to reveal that we have obtained total 541 registrations from various states of India, UP, West Bengal, Odisha, Jharkhand, MP, Uttarakhand, Tamil Nadu, Telangana, Maharashtra, Haryana, Gujarat, etc. And other countries including Saudi Arabia and Nigeria too. We have also received almost 10 on-spot registrations even today. Almost 309 attendees joined us today to attend the workshop offline. I am glad to share that we have received almost 141 abstracts for oral presentations, which was held during the post-launch session yesterday. That is on our second day of AA 2023 from 2 to 5 p.m. Parallelly, the session was conducted. The selected review articles are done in Zoom platform. For the research article, we have selected another platform, that is the Google Meet. The selected areas are pharmacology, pharmaceutics, pharmacochemistry, pharmacognosy. The prize money will be awarded along with the winner certificate to best presenters in each of these sections today on stage at the end of this ceremony. One thing that I want to express here before the audience, and one those are attending this in online mode, based on the feedback obtained from our delegates, even from our national and international speakers, we are definitely going to plan and carry out similar kind of events in future in a more majestic way. With these words, I will once again congratulate and extend my appreciation to my whole team members, the organizing team members, my faculty members, on my dear students, student volunteers, and definitely all my delegates, those who have joined me both in online and offline mode. Without you, this is not possible for this in the grand success of this event. So from AIP, AUP Lucknow campus, definitely I said that we are organizing this international conference come workshop on a very topical theme, and this topical theme is leading to this grand success of this event. Thank you one and all for your patience and joining us. Now I would like to request Dr. Husni to please proceed.
Thank you, ma'am. We are really blessed to have our Honorable Pro-Vice Chancellor, sir, Wing Commander Anil K. Tiwari, sir. So I will just request sir to join us on stage and bestow our audience with his words of blessing, because your very presence is very inspirational for us, and your words have always been inspiring us and keep on motivated forever. So, sir, please, can we have you on stage? And I would request to welcome sir on stage with a great round of applause.
Respected chief guest for this valedictory session, Dr. Sandeep Chaudhary ji. A very warm welcome to Amity University, Lucknow campus. Dr. Anuradha Mishra, all my esteemed colleagues, it's my privilege to be here to see the success of this international conference in workshop mode. That is alternatives to animal experiments, exploring imminent possibilities in preclinical drug discovery. Like Professor Anuradha said, they had a huge success with this international conference in terms of number of participants, in number of abstract they received. So my big compliment to you for doing so well. And it's my wish also that we continue with this particular topic in near future, because this is so relevant and this is so apt, not only from ethics point of view, but also on the viewpoint of sustainability. If we have to sustain research, if we have to do cutting-edge research, then it's just not possible to spend hours and hours on experiments alone. The time that we spend on experiments, there has to be a better mechanism to cut it down. And there's a lot to explore. We were just having a discussion over the lunch table as to why not stem cell technology is now capable of almost creating any organ or organelle on a chip that could be utilized to study any kind of an effect. Using the stem cell technology, it's possible now to generate any animal organ or organelle on the chip. And with a lab on the chip, you can almost do any experiment theoretically, provided we use advanced computational tools, because it's very, very difficult to mimic, simulate, and understand biological and pharmaceutical processes. They are one of the most complicated things. But then the rate at which the tools are expanding and the rate at which you have shown enthusiasm, I think everything is possible. If we are to overcome The tragedies like pandemic, which have taught us perhaps 3 years is too long a period to create a vaccine. If we're going to create them too fast, if we're going to have a preventive methodology coming very fast, then the only answer is the digital and the computational tool. Because using this digital and computational tool, you can actually create a digital heart, you can create a digital brain, you can create a lung of any animal or a mammalian specimen. So that's the kind of the possibility which the future holds. It's very interesting, it's very promising, and at stake is humanity, it's well-being. So when something so high is at stake, nothing is impossible. So my big compliments to each and every one of you present over here, participating in this valedictory ceremony. I hope you'll take this message home and come back next year with greater vigor, greater appetite for learning. All the best. My good wishes to each and every one of you. Jai Hind.
Thank you, sir, for your words of confidence and appreciations. They really mean a lot to us. It is now time to encounter some of the feedbacks from our attendees, online and offline. And may I first have one of the eminent and enthusiastic speaker from the International Forum, Dr. Tavinda Hassan. She has been attending all our sessions so enthusiastically for the last 3 days. And may I now request the IT team to make her visible on the screen so that we can have an interaction and have a feedback from ma'am. Meanwhile, let me remind you all that Dr. Tavinda Hassan is presently working as an associate professor of human anatomy, histology, and embryology, College of Medicine, Prince Noor bin Abdul Rahman University, PNU, the Kingdom of Saudi Arabia.
Yeah. Hello, good afternoon, everyone. And
Good afternoon, ma'am. Thanks
yes.
for joining, ma'am, and I request you to share your feedback and your suggestions to make improvements also.
Yeah, first of all, I was very much impressed by this conference, and I gained a lot of new ideas, and I was really impressed by the speakers as well as the involvement of the students. Like, I was very happy to see such a huge number participating, because usually, like, I attend a lot of conferences and I don't see that many participants, but here I see lots of participants, and also they were eager to ask many questions and intelligent questions, and that shows a lot of proactive engagement on part of the students. So this is something that's very impressive. And the way this conference was organized, if I compare it with international conferences I have attended a lot as a speaker in UK, in US, in Europe, even I went to one in Harvard and I went to one in Royal College. So I'm comparing from those standards. So I will really say the truth that India is nowhere less, not at all. And in fact, I would say that the way it was organized and the richness of the content, that was more than those places. So it was actually a kind of a surprise for me, because usually I'm not so much into Indian conferences, but this conference really changed my viewpoint, because maybe I was comparing back when I was a student. So I was a student in 1995. I completed my MD in 2003, and this is 2023. So I was living with those impressions. I was not so much engaged or involved in India in that way. I was more into the West and the Middle East. And joining this conference was because of a close contact of mine that I felt the motivation to. I said to myself, okay, let me join, let me see. And I am coming with the mindset bug that I had in 2003. So imagine, like, it was a surprise, a very pleasant surprise. And I felt so proud of it. Like, this is our India, this is our university, and wow, look at the way they are organizing, and look at how they are bringing the speakers, the topics, look at our students, the questions that they are asking. When I compared, I said, this is actually better than those. And believe me, I'm not lying. I'm just saying something that's from my heart. And I feel so proud of that fact, because we have come a very long way. And I am noticing a remarkable difference, because when I compare like 20 years ago, like from a mindset of a person who has been, let's say, a decade away from the Indian education system. So, and I am, so-called impressed by the West and these places. But then I see, wow, this is like, it changed my perspective, and we have gone like really, really, we have achieved a lot. And I really want to congratulate Amity University, especially your college, the way it was done. It left a lasting impression, and I would, I would really want to participate again in any future conference. Thank you so much for giving me the platform to express my opinion, like my very honest views.
Thank you, ma'am. We're extremely thankful and overwhelmed with your feedback and your suggestions. Also, will look on to for while we are planning for such miraculous events in the near future.
Yes.
And I must also thank and appreciate for your dedication, because whenever we have opened the Zoom platform, the first thing we have got clicked on to was Dr. Tavindra Hassan. So I'm extremely thankful for an active participation from your end also, ma'am, and we are definitely looking forward to have you as in person in the university campus very
Yeah.
soon, whenever you are planning for this. Thank you,
Thank
ma'am.
you. Thank you so much.
Feedback are definitely required for even improvising answer. So we do promise that whatever lacuna has been there, we are definitely going to work on the same to improvise the standard of the conferences and workshops that we are planning to undertake in the near future. So now may I call upon the stage one of our attendees, those who are present with us today in the offline mode. That is Umar Muhammad Faisal, if I am not pronouncing the name wrongly, from Integral University. He is from Nigeria and he is one of the participants. So I will request Umar to speak to you.
Good day to you all. My name is Umar Muhammad Faisal, and I am a fourth-year student of Integral University. I am a Nigerian. First of all, I would like to show you how impressed I am. This conference has really beaten my imagination, ranging from the number of participants I have seen and our eminent speakers, our invited guests who have, out of their busy schedules, managed to be here with us right from day one to the day 3. I would like to send my sincere appreciation to you all. Secondly, I would like to commend the effort of the organizing committee. They have really done a wonderful job. And I wish to see more of this in the coming future before I leave India. Because my stay in India for the past 3 years has given me the opportunity to attend so many conferences, but I can say by far this is one of the best I have attended so far. So congratulations to Amity University. And this conference has really broadened my understanding of the pharmacy field actually. And it has shown me ways or the path to take after my B pharmacy. And I'm sure I will keep the connection with Amity University even after my graduation. So as we come to the end of this international conference from workshop, I would like to wish you all a safe return to our respective destination. Thank you very much, and I really appreciate it.
Thank you, Umar. May I now have on stage Rabia Ahsan from Integral University again. She is a PhD scholar and she has keen interest to share her feedback on the stage.
Good afternoon, all of you. Myself, Rabia Ahsan from Integral University. I feel privileged to be a part of this conference come workshop, and it was very fantastic experience and valuable lectures from honorable guests and speakers like Dr. S. Anupmani, Dr. Tabinda Hassans, Dr. Muhammad Arshan, Dr. Yasir sir. They provided valuable lectures about stem cells, dockings, zebrafish, etc. And thank you so much, Professor Dr. Radhakrishnan and all coordinators for organizing such an interesting and valuable conference come workshop on AEE. Thank you so much.
May I request now Nitin Rajan, faculty from the Government College of Pharmacy, to share his views. Do we have Nitin Rajan? Okay, so with that, there is the list of, feedback is floating all around. We are now at the end of today's ceremony, and now I will request all the dignitaries to again kindly join us on stage because it's now time for appreciating some of our student participants and student volunteers. So can I have all the dignitaries on stage, please, sir? The dignitaries, Director HR, Asita ma'am, our beloved Professor Dr. Radha ma'am, to please join us on stage for distributing the certificates to the winners of the oral presentation competition that we conducted during the second hour of yesterday's session, that was from 2 to 5 p.m. online. So it's now time for distributing the certificates and declaring the results of oral presentation competition for yesterday's session that was conducted online. We have received almost 141 abstracts for oral presentation competition, and out of that, selected abstracts have been segregated to both review and research sections for 5 different sections. So let me read out the names of the winners for first and second positions of pharmacology section. The first position has been occupied by Bhagya Bhumi Saab on the title Unraveling the Mechanistic Role of an Alkaloid and Recovering Phytoconstituents of Ether Maltose in Mitigating Inflammatory Bowel Disease. See it online, sir. But can we have a great round of applause for the team? The second position has been occupied by Siddhi Srivastava from Department of Pharmaceutical Sciences, Baba Saheb Bhimrao Ambedkar University, Vidya Vihar, Raebareli Road, Lucknow, MC online. Yes, Siddhi. And her research area was deciphering the link between vitamin D receptor and chronic stress-induced male infertility, a promising pathway to repurpose FDA-approved drugs through in silico analysis. So may we have a great round of applause for Siddhi for securing the second position, and she has been awarded with a prize money of rupees 1,000 for attaining the second position in the section pharmacology original research. Congratulations, Siddhi, and thanks for joining us offline. I will now firstly read out the names only of the winners because they are all online. From Pharma Chemistry, the first position has been secured by Jivan Parker from School of Health Sciences and Technology, University of Petroleum and Engineering Studies, Uttarakhand. Second position being bagged by Ketki Rani from SGT College of Pharmacy, SGT University, Gurgaon, for Pharma Chemistry Original Research Articles. From Pharma, can we have a big round of applause for Jivan? Let me tell you that the best presentations for first and second positions are being awarded with a prize money of rupees 1,500 for the first position and rupees 1,000 for the second position. So now, let me have the privilege of announcing the winners for the first position from Pharmaceutics. That is Jagriti Desai from Raman Bhai Patel College of Pharmacy, Shantor University of Science and Technology. And the second position being occupied by Vinit B. Agnihotri from SDKX Institute of Pharmacy, Jule, Maharashtra. Can we have a round of applause for the winners, please? Moving on to pharmacognosy, the first position has been occupied by Kadam Apeksha from Faculty of Pharmacy, the Maharaja Sayaji Rao University of Baroda, and second position being occupied by Alju Vera from Department of Pharmaceutical Sciences from Prayagraj. Moving on to the review section, we have the first position being occupied by Sinha Nita from Faculty of Pharmacy, the Maharaja Sayaji Rao University of Baroda. And second position being occupied by Deepak Mishra, Maharana Pratap College of Pharmacy, Maharana Pratap Group of Institution, Kanpur. Can we have a big round of applause? Let me tell you, sir, we could reach to the grand success of this event only because of the constant and dedicated support of most of our students from Pharmacy and Farm being asked to be volunteers. They have worked day and night, hands in hands with the faculty members to make this event a grand success. So I will just request all of you to come onto the stage. I'm reading out the names. Adarsh Singh, Amrit Bharti, Silpi Upadhyay, Nigar Fatima, Sara Saeed, Mohammad Kaif Khan, Pallavi Malik, Ashish Roshan Singh, Chandrasekhar Nishad, Abhishek Yadav, Saumyadeep Roy, Satyam Singh, Priyan Singh, Ankit Kumar, Amit Singh, Roshan Kumar Singh, Khushbu Singh, Ankit Singh, Soeb Ahmed, Vishwajit, Ayushi, Simran, Utkarsh, Gupta and Utkarsh Upadhyay, Triveni Mishra and Tejas Patni. All these students have worked day and night, even according with their classes and all. They have helped us a lot. So I think there should be a great appreciation to all our student volunteers. And I request the honorable chief guest and the dignitaries to kindly hand over the certificates to the students. I am extremely sorry that I have skipped names. Sisti, Anjali and Vatsalya, they were the MC for the first inaugural ceremony of this event also. So I request Sisti and Vatsalya to also join us on the same. And can we have small class and then request the students to leave the stage also. Thank you, sir. We can now, I would now request you to take a quick off stage. I would request all the dignitaries to clear off stage. It's now time to bid goodbye. So with a mixed feeling, like a bit of joy and a sadness also. Bit of joy because we have successfully completed and conducted this international conference from Warsaw on a hybrid mode. And bit of sad because we are now not able to meet you all people, those who are attending online and offline this particular session. So may I now take this privilege to offer the official vote of thanks along with all my co-organizers, Dr. Rahul, Dr. Yasir, and Dr. Santha. I request all of you to kindly join me on stage so that I can sit out and offer the official vote of thanks to close this ceremony. I stand here today as one of the proud and grateful organizing secretaries along with my fellow mates, Dr. Prathamis, Dr. Rahul Maurya, and Dr. Mohammad Yasir to express my heartfelt thanks to all those who have made this event a grand success. I would like to start by thanking our beloved Director, AIP AUUP Lucknow Campus, Professor Dr. Anuradha Mishra ma'am, who have always been supportive for all our initiatives and encouraged us to strive for excellence ever since we conceptualized this grand 3-day-long event on hybrid mode. Words are not enough
Words are not enough to thank your constant guidance and support to shape this international conference and workshop. Thank you, ma'am. I thank all honorable dignitaries, including esteemed Provincial Sir, Dean Academic Sir, Kamar Madam, and the honorable speakers, Dr. S.K. Rao and Dr. S. Abu Mani, for taking out time for their extremely stringent schedule and graced this ceremony with their presence. An event of this dimension cannot happen overnight. The wheels start rolling months in advance. We have been fortunate enough to be backed by our organizing team of very motivated and dedicated faculties and students, volunteers. I thank everyone enough for the involvement they have shown and the willingness they have expressed to take on the completion of tasks beyond their comfort zones. The IT team deserves a special thanks for looking into the proper convenience of this program online. Thank you, Gaurav Mathu sir, Pragyan sir, and Pragya madam for the same. I also thank Sauji to capture this event in camera and convert it to memorable moments. Extremely thankful to Deputy Dean Director, Admin, the DN Singh sir, and team for amazing the food, other arrangements for smooth conduct of the event. I thank all attendees attending this event online and all our students for PharmD and M Pharmacy attending this online. Thank you one and all. Thank you very much. Okay. I request you all to stand up for national anthem, please.
नहीं चल रहा उससे नहीं चल रहा बेटा Thank you all. Or announcement. Students, please wait for some time. So we will have a group photograph. Participant, please wait for a few minutes. We will have a group photographs. हाँ गई का पेनड्राइव मेरे से पेनड्राइव
नहीं
अरे लोग पेनड्राइव दे दिया
May I request all the dignitaries, the faculty of AIP and the student volunteers to join us on stage to have a snapshot.
दे दिया हाँ निकाल लीजिए निकाल सीधे आराम से भाई आराम
I request all the faculties who are walking behind stage.
चलो बुलाओ
I request all the faculty those who are even walking backstage to be gathered in the auditorium for a final snapshot. The attendees who have joined this event since the last 3 days online. I thank you all to be a part of this event. Shortly, we are going to share the feedback link for today's session, which is mandatory to be filled up. You must fill the feedback before 5 p.m. today. The feedback will be shortly shared with you. I once again extend my deepest sense of gratitude from AIPKU Lucknow to be the part of this event and making it a grand success. So thank you, the online attendees. We are now setting this international conference from Warsaw. Thank you all. I also request and invite those participants to be with us on stage for the snapshot. If someone wants to join us, they are invited.
पहले आप लोग पहले आप लोग वो नहीं है चले गए आ जाओ तो नीचे भी हो गया तो बड़ा ही बच गया ना बाहर बाहर क्या है गोटी रंग रहे तो आदत पड़ गई क्या फोटो खिंचाने नहीं देखा है हम लोग कभी आ जाएगा पता है और आदर्श और एक मैं आ जाऊँ क्या आइए भी डालना है मेरी भी अच्छा